Hello everybody and welcome to Draminor Medicals. Today we're going to take an overview of the cell, okay? So why we're doing this is because when you start to dive deeper into biology and physiology, biochemistry, microbiology, pharmacology, this picture right behind me is going to be very important. You have to know the big picture, okay? A lot of students, they study the nucleus, right? Or they study, let's say, biochemistry, molecular biochemistry. And they're studying translation, transcription, but they do not know where all of this is going on. They don't know how the messenger RNA, for instance, goes from the nucleus and reaches the endoplasmic reticulum, for instance, okay? Or they don't know how the mitochondria intertwines in the cell itself, in the cell processes, okay? So let's talk about the structures behind me on the whiteboard really quickly. So here you've got the cell membrane. A cell membrane simply creates boundaries of the cell. Now I am standing in a room. I cannot have a room if it did not have any walls, right? So think of the cell membrane as the wall of the cell. It's what gives the cell the structure. Without a cell membrane, you have no cell, okay? So number one, cell membrane would be this portion right here. Now, a very important thing about cell membrane is that the cell membrane itself, if we take this right here and you look at it, it's called a phospholipid bilayer. Why is it called a phospholipid bilayer? Well, that's easy, because it's made out of phosphate and it has lipids, and we've got two layers. So phospholipid bilayer is, well, I mean, it's self-explanatory, right? You've got a phosphate head, you've got lipids right here, and you have the inner layer, layer number one, and layer number two. And therefore, we see a phospholipid bilayer. Understanding the phospholipid bilayer is very important. A lot of students simply think that the phospholipid bilayer is only present in the cell membrane. That's simply not correct. The phospholipid bilayer is present in many structures inside the cell, and it is very important. That's why we're going to spend quite a little bit of time on it, understanding it, breaking it down. In fact, we're going to make a whole series on the cell membrane. But for now, just know that the phospholipid bilayer is present in these, or it makes the cell membrane. However, the phospholipid bilayer also makes the layers of the nucleus. And by the way, if the cell membrane has layer 1, 2, has 2 layers, the nucleus has 4. The mitochondria also has 4, okay? Golgi apparatus has 2 lipid layers. I'm not saying that there are 2 lipid bilayers, I'm saying 2 lipid layers. So 1 phospholipid bilayer for the Golgi apparatus. For the endoplasmic reticulum, same thing. It has 1 layer, a single layer of phospholipid bilayer. This is the peroxisome, the lysosome, okay? So basically, the idea is the cell membrane, or not the cell membrane, sorry, the phospholipid bilayer is not only present in the cell membrane. That would be incorrect, okay? So get your facts straight. Phospholipid bilayer is present in the cell membrane, is present in the nucleus, it's present in the endoplasmic reticulum, in the Golgi apparatus, in the mitochondria. In fact, we have a name for them, for the structures that have a phospholipid bilayer. And these are called membrane-bound organelles. Okay, now before we go there, let's talk about some definitions. Number one, protoplasm. Now, first of all, you have to understand that protoplasm is an outdated word. It is no longer used. However, you may, hear it once in a while being thrown around here or there, but protoplasm is not a word anymore. Why? Well, basically, the origins of protoplasm is when a Czech scientist, 1839, 1840, he took his primitive telescope, looked under it at a cell. Okay, so basically he couldn't see the nucleus, the peroxisome, the Golgi apparatus, all of these structures. He simply saw, a substance. And so he said, you know what? This substance is going to be called a protoplasm. And that's where the word originated from. Now, come to the 1930s, 1940s, where we had a little bit more advanced telescopes, let's say. And the scientists, they kept the word for some reason, okay? But at that time, they started to distinguish the nucleus, the mitochondria, the Golgi apparatus, etc. But they kept the word. Now, as science, of course, advanced, we were able to make better microscopes, And eventually the protoplasm was not a weird anymore, because protoplasm, by definition, simply means all of this. So all of this here is going to be called protoplasm. That does not make sense. And therefore, we came up with the weird cytoplasm and nucleoplasm, nucleus, cytosol. We came up with all of these, okay? So what is the cytoplasm? Is it the same thing as cytosol? And I only bring this point up because a lot of students make the wrong assumption that the cytoplasm is the same thing as a cytosol. That's incorrect. You see, a cytoplasm encompasses four things. It encompasses the cytosol, the inclusion bodies, the cytoskeleton, and the organelles. Okay, so a cytoplasm or the cytosol is simply one component of the cytoplasm. So let's talk about the cytoplasm, shall we? What is the cytosol, first of all? Cytosol simply refers to the aqueous or the fluid that is going to be present inside the cell. Okay, this is where cellular processes take place. Okay, and this is called the cytosol, right? It is not the cytoplasm, it is the cytosol. Again, I know I talked your ear off about this, but it is very important for you to make the distinguish between them. Because when a messenger RNA goes out of the nucleus, some books even, they say that the messenger RNA, the mRNA, goes out of the nucleus into the cytoplasm. Again, that's incorrect. It goes outside of the nucleus into the cytosol. So what's the cytosol composed of? Well, mainly 70 to 75% of it is made out of water, H2O. That's simple. It has potassium ions, you know, so potassium, chloride, sodium. It's got enzymes. It has ribosomes in it, in fact. Yeah, so sometimes you might find ribosomes just floating in it. What else does it have? Well, it has the inclusion bodies in it. So cytosol is, of course, because it has these enzymes and these dissolved proteins and these ions in it, it's kind of jelly-like material, really, so it's not really fluid, okay? And therefore, some textbooks refer to it as a jelly-like substance. That's the cytosol, again, not the cytoplasm. Okay, so number one is the cytosol, done with that. Number two, inclusion bodies. What are inclusion bodies? Inclusion bodies can be things like glycogen droplets. And these are present abundantly in the muscles. Why? Because muscles, a lot of the times, they deplete their glucose, they need glycogen. Where do they get it? From the inclusion bodies. Okay? Liver cells, for instance. Liver cells, the liver is where fat is synthesized. And therefore, they're going to have lipid droplets. Again, these are inclusion bodies. Sometimes pigments also are present as inclusion bodies. And of course, the inclusion bodies, put in mind, do not have a membrane around them. Okay? So that's number two. Number three, the cytoskeleton. Now, again, with the wrong assumptions. The cytoskeleton is not going to be present only for structural support. I mean, that is one of its functions. And a lot of students, again, make the mistake of thinking that the cytoskeleton is only going to be present underneath of the cell membrane. Now, while that is not completely wrong, it's not correct either. Why? Because the cytoskeleton is divided into three things. You've got the actin core, or the microfilaments, right? Then you've got, for instance, this. This here is called the intermediate filaments, and it's usually found around the nucleus and some other organelles. Okay? Number three are the microtubules. And we're going to talk about these when we talk about the cell cycle, about the mitosis. However, for now, for this lecture purposes, just know that the cytoskeleton is made out of three components: the microfilaments, the intermediate filaments, and the microtubules. Okay? Number four are the organelles. So we're talking about things like the mitochondria, like the lysosome, like the Golgi apparatus, like the peroxisome. The nucleus is an organelle. However, when we talk about cytoplasm, the nucleus is not included. Just put that in mind. Okay? So to recap really quickly, we talked about the cell membrane, and we said that a lot of the organelles are going to be covered by a phospholipid bilayer, not just the cell itself. Okay, that's number one. Number two, we talked about the protoplasm. We said it's an outdated term and that we do not use it anymore. Okay? Number 3, we talked about the cytoplasm and the cytosol, and we talked about the differences between them, or not the differences, we just want to make sure that you know and you understand that the cytoplasm is not the same thing as the cytosol, that the cytosol is simply part of the cytoplasm. We also talked and we said that the cytoplasm has 4 components, and this is the cytosol, remember the fluid part of the cell, the inclusion bodies, the cytoskeleton, and the organelles. Okay? Now, taking a look, of course, at the organelles, again, endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, peroxisomes, etc. Now, the nucleus itself, you should be also aware of different nomenclatures, okay? So what's the difference between the nucleoplasm and the nucleolus? Or what is a nucleolus? A nucleolus is simply a structure that is going to be present in the nucleus. It's called nucleolus. Is it always present? No. The nucleolus is not always present. Why? You see, the nucleolus is, it's the site of ribosomal RNA synthesis. That's all the nucleolus is. Nucleolus doesn't have boundaries. It's not bounded by anything. A nucleolus is something that we see during interphase under a microscope. We see it because the chromosomes at this stage, during interphase of cell cycle, are bundled together so tightly that they appear under the microscope. That's the nucleolus. Nucleolus is not a permanent structure. Mostly, it's not present. What's the nucleoplasm? Well, nucleoplasm is synonymous to the cytosol. Remember, we said the cytosol is the fluid portion of the cell. Well, the nucleoplasm is the fluid portion in the nucleus. Okay? So again, definitions, know them, they're really important. MBO, or membrane-bound organelles. You see, we have many organelles. We have many organelles in the cell. Most of these organelles are going to be bounded by a membrane. What does bounded by a membrane mean? Well, it simply means that the organelle has a covering of phospholipid bilayer on it. Therefore, we call them membrane-bound organelles because they are bounded, they are separated from the cytosol, again, not the cytoplasm, from the cytosol and from each other, of course, by a membrane. Okay? And therefore, their names are membrane-bound organelles. Now, membrane-bound organelles are going to be things like the mitochondria. Remember, we said it has 2 double phospholipid bilayers, so it has 4 layers. The nucleus, again, it has 4. Endoplasmic reticulum has a single layer of phospholipid bilayer, meaning it has 2 layers. The peroxisome, the same thing. Golgi apparatus, the same thing. And the lysosome, the same thing. Now, there are other, of course, membrane-bound organelles. We're not talking about them because this is an overview. Okay? We're only going to talk about the structures that are usually, or let's say always present in the cell, no matter what stage it's in. The only exception here was the nucleolus. And I did that because, again, clarity, okay? Now, how do these structures interact with one another, okay? And to make things easier, we're going to start with, let's say, insulin, right? So what happens is, a signal comes, passes through, and goes to the nucleus. This signal could be growth hormone, it could be a growth factor, it could be a stress stimulus, whatever, right? So it goes to the nucleus and says, make me messenger RNA for insulin, for instance. Let's say it's insulin, right? So the nucleus goes to the DNA, there's a specific gene on the insulin, or for the insulin protein, sorry. It opens it up, it opens the DNA. The DNA is read. We're not going to get into detail. Again, this is an overview. I just want to show you how all of these intertwine with one another, how they work together. So you've got the messenger RNA. The messenger RNA is formed inside the nucleus. It goes out through the nuclear pore complex, right? And now the mRNA, the messenger RNA, is going to be present here. So mRNA. It goes into the cytosol. Okay, now in the cytosol, the messenger RNA is going to bind with a ribosome. Let's make the ribosome larger, just, so you can get the idea, right? So the messenger RNA binds to the ribosome, and since it's insulin, okay, we're not going to go there, okay? It's probably too advanced now. But anyway, the ribosome grabs the messenger RNA and takes it to the endoplasmic reticulum. Now, on the endoplasmic reticulum, it starts translating it, right? So it starts to make amino acids, and they're like blocks, and they're put in next to each other. At the end of this, you've got a polypeptide. What's a polypeptide? It's simply many amino acids or many proteins that are hooked together, right? Now, they enter into the lumen of the endoplasmic reticulum. What happens there? Well, it's protein. So it gets folded, probably some sugars are added, doing endoplasmic reticulum stuff. Again, we're not going to go into detail. From the endoplasmic reticulum will bud off and give a vesicle that has the protein in it, right? So again, a signal came, we gave you a messenger RNA. The messenger RNA, through the nuclear pore complex, went outside into the cytosol. In the cytosol, ribosomes grabbed onto the mRNA, took it to the endoplasmic reticulum, and started, doing its thing. It started to translate it. After that, went inside the endoplasmic reticulum. Endoplasmic reticulum, it probably folded it. It maybe added some sugars to it, whatever. And it released it. Now, where does it go? Well, what happens is actually that it goes through this structure, which is called the microtubule. Okay, again, look how everything is coming together. From the microtubule, it then is going to pass and go to the Golgi apparatus. Of course, the Golgi apparatus, there's a cis phase and there's a trans phase, but we're not going to talk about this right now. Okay, remember, we're only doing this because we want to relay the information that everything's important. Anyway, it goes to the Golgi apparatus, step 1, 2, 3. Eventually, it gets released like this, right? And it goes up, up, up into the cell membrane, right? And it fuses with the cell membrane. Something called exocytosis happens, okay? So basically, the vesicle goes, when it reaches the cell membrane, it fuses with it, and then on the outer surface of the cell membrane, it opens and insulin is secreted in the extracellular environment. Extracellular environment meaning any environment outside of the cell, right? So it went up, 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 came here, then, it opens up like this, and the insulin is secreted outward. Now, the mitochondria, of course, is going to give ATP, it's going to give adenotriphosphate to all of these cellular processes so that could take place. Put in mind, of course, mitochondria doesn't only generate ATP. I mean, if I was a mitochondria and that's all I was known for, I'd be mad, because the mitochondria does a lot of things. Okay? So basically, this was the cell overview. In the next lecture, we're going to talk about the cell membrane. Okay, we're not going to be talking about the cell membrane. We're going to talk about the phospholipid bilayer. Why? Again, remember we said that the membrane-bound organelles contain a phospholipid bilayers. Some of them have even double phospholipid bilayer. Okay? This was the end of today's lecture. Have a good day.